Welcome to the Cucurbit Working Group. I'm Dr. Richard Hassel, and I'm chair of the Cucurbit Working Group. Today we're going to discuss many of the accomplishments that we have done as a Cucurbit Working Group. The first one I want to talk to you about is the development of new rootstocks. Under the direction of Dr. Pat Wechter, Clemson University, as well as the U.S. Department of Agriculture, have released a new rootstock called Carolina Strongback. It is now licensed to the Syngenta Vegetable Seed Company. This slide shows you the high level of resistance it has to a number of soil-borne and foliar diseases. This rootstock has been tested throughout the United States, as well as Mexico and Australia. This rootstock not only works for watermelon grafting production, but also works for cucumber grafting production, both greenhouse and field production, if any of you are interested in that. With the cucumber production and vegetable production, it also enhances early set of the cucumbers, which is something that we have not seen in watermelons before. So I think there's some potential there for that rootstock on the cucumber side. In addition, we have been working and are still working with the process of 5 other rootstocks that have advantages and disadvantages to our current release of Carolina Strongback. We've been using each of these in crosses between Carolina Strongback to improve both the horticultural characteristic of Carolina Strongback as well as the development to further foliar diseases. If you're interested in getting some of these seeds, myself and Dr. Sandra Branham have an MTA agreement with USDA. And this last year we did a seed increase of each of these lines. And we have plenty of seed on hand to disperse additional availability of these rootstocks to any of you that might be interested. In addition, we've also been working with melon rootstocks to increase both production as well as disease packages by using grafted melon rootstocks. Here are 3 melon rootstocks that we think have a lot of potential, but of those 3, we also have an additional 2 others that we still are out in judgment on as to whether they'll be successful or not. Their disease packages are listed in this slide, as well as the amount of seed production expected per acre after each one of these. Again, we have ample supply of seeds for these since we did an increase on those as well this last year. Dr. Shaker Cusick, also at the USDA, has been doing a lot of breeding with the Cucurbit, Marshada, and Maxima rootstocks for resistance to Phytophthora. Of those, he's been screening both the 319 lines of Cucurbit and 512 lines of, Marshada, the 319 lines, PI lines of Marshada, and the 510 lines of Maxima, screening for resistance to Phytophthora. Of those lines, he has found out of the Maxima 2 lines that have developed out of that, and a new source of resistance to crown rot Phytophthora is found in the Marshada, where he has found 12 new lines for development. These advanced lines are now being used to develop hybrids between Marshada and Maxima, hybrid rootstocks for watermelon and melon production. He's also been looking at the development of powdery mildew and the resistance to powdery mildew. And he has 3 rootstocks onto the bottle gourd side that have a lot of potential that show complete resistance to powdery mildew. With this, he has found that these powdery mildew is also transmitted across the graft union, as we are seeing with a lot of other foliar diseases, but in this one in particular, powdery mildew, grafted on this rootstock to a very susceptible powdery mildew cyan material has complete resistance to powdery mildew. Shows a lot of promise to us in that a lot of the foliar diseases can also be controlled by some of our rootstock material. I'd like to talk to you now about a little of the horticulture characteristics and making sure that we provide the industry with a consistent, reliable rootstock and cyan material to graft. This is important because in order to develop a proper grafted plant to develop into the industry, into the field, a consistent, reliable, very good grafted rootstock material, grafted material to put out in the field is essential to evaluate any prospects that grafting works. So we've been doing a lot of work with doing inside grow rooms, controlled environments, so that we're not relying upon a greenhouse environment which changes from day to day depending on the daylight outside as well as the temperatures. And so we've been looking at light duration, quality of light, effective temperatures on that seedling development. We've been trying to develop what we call a cookbook that we can supply that information consistently, and so we can develop a high quality transplant for the open market. And we're doing that within the grow rooms themselves. This just gives you an idea of the effect of what we call the amount of light duration across the bottom here with the temperatures. And this red line across the middle shows that that's the ideal height that we'd like to have. So if you're looking at cyan development, you're looking at temperatures about 70 degrees and at least 18 hours of sunlight. If you're looking at the watermelon rootstock, for example, Carolina Strongback, as this one is, you want a little bit longer hypocotyl length. And so you're looking at something at 40 millimeters in length, but you're looking at a higher temperature in order to control that, to get that kind of length. But you're looking at 21 hours of light, 21 hours or greater amount of light at 70 degrees. And then you can, we'll get you that on a consistent basis. The interspecific hybrid squash, which grows really quickly, rather quickly, has the same type of effect as temperature and light. And if you look at 75 degrees there, and if you grow across the line there, you can see that you can get that between anywhere between 18 and 24 hours of light. Now I'd like to talk to you a little bit about what Jonathan Schulteis is doing at NC State. Jonathan Schulteis is an excellent horticulturist, and he's been evaluating many of the commercial available rootstocks and determining their effects on plant growth and vigor and fruit yield and quantity of both mini and standard sized triploid cultivars. He hypothesized that certain rootstocks would specifically matter with cultivars and fruit size and basic on effective cultivar and rootstock. Regardless of the rootstock, he found that the early growth and yields were gained with minimal and standard sized melons was not challenged with disease conditions, would compare with the non-grafted control. In other words, really grafting had little benefit to be gained by growth and yield. Also, the goal was to determine how cultivars reacted with the impact on firmness in watermelon flesh and the incidence of hollow heart. With the interspecifics, firmness watermelon flesh, the flesh is much more firmer, but with less hollow heart, and that was based on cultivar and rootstock species. The interspecific hybrid rootstocks had much firmer flesh and less hollow heart than did the bottle gourd rootstock. He also had the goal of compare root growth with each of the rootstocks. pumpkin squash and the interspecific hybrid squash produces more total root length, much more surface area than did the bottled gourd or the actual or the wild watermelon. They're very, very aggressive rootstocks. And as all of you know, the watermelon rootstock itself is not a very vigorous rootstock, and that's why it gets attacked to a lot of the diseases we're going to talk about in a minute. But the interspecific hybrid rootstock is very, very vigorous rootstock, and it holds up very well because it produces a lot more surface roots as well as the tap and below ground root systems. Zin Ho at University of Florida has done a lot of work with grafting. She's been excellent in using various cucurbit rootstocks to control fusarium wilt and watermelon production. As you know, Florida is quite heavily infested with fusarium. And she also wanted to look to see, because of the cost involved in grafted plants, whether we could actually reduce the amount of grafted plants needed per acre. And so she did density studies as well as to maintain fruit quality and yields. And then the third objective was to see if she could promote earliness using grafted plants, could actually increase yearly yields as well as increase overall yields. First one I want to show you is the bottom graph here. The inoculation of fusarium in this area where she grafted Melanie onto the cultivar Melanie was grafted onto the rootstock Super Shintosa, which is interpacific hybrid squash. You look at the other two where the incidence of fusarium is. This one had fusarium which was inoculated, but there was no fusarium located at all. In other words, Super Shintosa is completely resistant to fusarium wilt. She also looked at the effects of fusarium wilt on marketable fruit quality. And she also showed that in this next study that not only is it resistant to fusarium, but the interspecific hybrid squash are very susceptible to root knot nematode and other nematodes that might be found in your soil, and heavily infested with galls. As we found over the years that both the interspecific and the bottled gourd rootstocks are very susceptible to the nematodes. In fact, more than anything else that we've ever seen, and much more than the standard watermelon rootstock itself. I mean, standard watermelon roots themselves. On plant density, this is where we've had some issue of controversy throughout our group. In some areas, the in-row spacing, we've seen that we could reduce the in-row spacing with grafted plants rather than going the 8x3 or 24 square feet per plant. We could actually go with 32 square feet per area per plant and not reduce yields, maintain the same yield and quality of our fruit. Other areas, we haven't been able to see that same effect go on throughout other parts of the country. So the debate on that is still out. She did the same thing she looked at with fascination watermelons. And she had different planting dates and densities for overall and looked at early yields as well as quality of the fruit. She did see some, however, some effects of off-colored or flavor to the watermelons. And that we'll discuss in just a little bit when we get into the post-harvest section. But overall, she found that grafted plants are much more effective in controlling fusarium, but she didn't see any earliness with grafted plants, actually more of a delay in grafted plants with earliness. Next, I want to talk to you a little bit about the disease section that we've really focused a lot on. And we have an excellent group of plant pathologists working on the various aspects of grafting. Two of those are Dr. Anthony Kyneth and Dr. Paula Aguilera from Clemson University, who have been working on the disease aspect of grafted plants for quite a while now. In this part of their talk, I want to talk to you a little bit about the goals and objectives. First of all, does root knot nematode increase fusarium wilt in watermelons? In other words, is there a effect of having one or the other, the increase of that possibility of having one or the other because of the one known disease? Also, does cucurbit rootstocks have resistance to Pythium? Pythium is becoming a real substantial problem in our area with watermelons, especially in the spring. And we're looking at ways of controlling Pythium also. In the first one, we haven't seen a correlation drawn at all between the root knot nematodes and the incidence of fusarium. They seem to work independently of one another. As far as the Pythium goes, we found four species of Pythium that have been detected in cool season, in warm season. And but we've also found that the interspecific hybrid squash rootstock seems to be totally resistant to Pythium. And that is shown by the yields here with the interspecifics. And they did much better than not having any control at all as far as the yields, both in the spring and fall. One thing that we were very disappointed in this study, the results of this study, is the results that we saw with Carolina Strongback. Carolina Strongback is a very, very vigorous rootstock. And if planted late into the summer months, it's so vigorous that the onset of fruit set is delayed tremendously. And so I think that's maybe the reason why you see a reduction in yields even over the non-grafted itself. Because in talking to Dr. Kynan's graduate student, it seems like that the Carolina Strongback definitely has resistance to Pythium, but it doesn't show in the overall markable yield and weight in this study. He has a number of publications that are out there. And if you'd like to read any of them and get a hold of anything, I'm sure Dr. Kynes would be willing to share any of these with you or point you in the direction where you can get a hold of those. Dr. Carol Miles at Washington State University is also another one of our chief horticulturists on this study, and she's been looking at verticillium wilt. That's a major problem out there in her area. And also she's been looking at some other characteristics besides verticillium, but mainly been focusing on the development of what we can do to control verticillium. She's found that grafted plants can control verticillium wilt. And she's found the interspecific hybrids do that, as well as some other incidents. They've also found that grafting, that there is no difference in fruit maturity or fruit quality due to grafting, except for hollow heart. And we've seen an increase in reduction in hollow heart, which we'll explain in just a little bit. She's also been looking at the same effects on cantaloupe grafting on cold-resistant roots, onto cold-resistant rootstocks, and seeing if she can improve the cantaloupe rootstocks. She hasn't had a chance to look at some of our new rootstocks that we've developed for melon grafting, but those definitely are going to be available to her to look at in her area for both melon as well as watermelon grafting. She has a number of publications that she's willing to share with you. So contacting Carol and to get a hold of those is a very good idea if you're looking at anything to do with verticillium wilt, as well as some grafting on 1-cotyledon grafting methods. And she's worked on a lot of other horticulture characteristics to make sure that we have a quality grafted plant. Josh Freeman at University of Florida is our root knot nematode expert on the Cucurbit working group, and he has studied a number of aspects of fumigation versus our grafting methods for controlling root knot nematode. Nematode management, first of all, you need to know the difference in where the profiles are for most of the nematodes. And so the objective was to look at that profile, evaluate resistant rootstocks, and evaluate soil fumigants and non-fumigant tools for those that are susceptible rootstocks. And also he's looking at viral management, evaluation of grafted watermelons under pressure from whitefly transmitted virus, which is becoming a very, very prominent problem in our area. So this gives you the idea of the distribution of nematodes within the soil profile. And you look at the majority of those is between 30 and 60 centimeters within the root knot, within the soil media depth. One thing that's very important to note from all of the studies is the interspecific rootstock as well as the bottle gourd rootstock are very, very susceptible to root knot nematode. He's also finding that the New Carolina strongback is completely tolerant to the nematode populations in their area. So that's one important thing to understand. Also, the virus transmission, he found no advantage to grafting as far as being able to combat cucurbit leaf crumple virus, leaf crumple virus that is becoming a very prominent virus in our area. So we have to look at alternatives to that, but grafting is not one of those that will take care of that. However, we are seeing some transmission across the graft union, and so maybe the next avenue is to find some kind of resistance to the resistance to a rootstock that might hold resistance to that virus that we could graft on, and we might be able to get some kind of tolerance across that graft union. Next, I want to talk to you about Dr. Penny Perkins' work with post-harvest physiology. Dr. Perkins is a leading plant post-harvest physiologist, and lately she's been working on taking on the real difficult task of trying to figure out what's causing hollow heart and how to correct hollow heart within our watermelons, a big task. But she also coordinates and heads up all the tests throughout the United States dealing with post-harvest. Grafting on different scion material and also different rootstocks poses a big problem because of that interaction with the grafted material. And so there's been a number of studies that she's been involved in throughout the United States, including ours at the Coastal Station of Charleston. For example, Zin Ho in Florida, she found some off flavors with rootstocks, especially with the Scross rootstock. She's found that there's a higher firmness quality. The fruit are much firmer with grafted. Lycopene's sometimes are higher with grafting. And there seemed to be no difference in century quality tests from our panelists with that. Also with Carol Miles, as I told you earlier, has done a lot of work. He's been working at watermelon quality on rootstocks testing. She has also found that flesh also became firmer. But also she found that hollow heart incidence also increased with grafting using certain rootstocks and certain scion materials. But in her test with hollow heart, had a graduate student that's just finished up her PhD working with trying to figure out hollow heart. And what they have found, that they've found definitely a decrease in hollow heart incidence with grafted material. And she's been examining both the pectin amounts and the fractions with the different acids within a watermelon, the cell wall thickness, measuring the different sucrose levels, trying to pinpoint exactly what's going on with hollow heart. To get a further explanation on that, I suggest you contact her graduate student, for further information concerning her work with hollow heart. But the interspecific hybrid, she's found consistently improved flesh firmness across most of all the scion materials. She says there's an increase in lycopene, citrulline, and Brix with grafted, but the effects are different with different locations. The incidence with having off flavor she has been pinpointed to the incidence of the flesh itself, and whether it is whether that flesh is associated with the pH. And she's found that the lower pHs of the fruit, the more the off flavors, and the higher the pH, 5 to 6, the sweeter the melons are. This is important because when you graft on the interspecific hybrid squash, the fruit will appear to be color-wise as far as red and very firm flesh. But as you take the pH of the flesh, you'll find out that it's on the acidic side, 5.3 down even into the 4s, and then you get your off flavors. Very, very interesting study. I'm sure she'll share that article with you that she has written on that paper, and it's very fascinating. Now I want to talk to you a little bit about weed control. Weed control is a big problem in watermelons, especially as watermelons develop in the early stages of the plant development. It's really critical that you keep the weeds under control. So Dr. Katie Jennings from NC State has been looking at weed control and looking at different aspects of weed control in certain weeds and controlling that. So her objective was to see if grafted plants would give her more of an advantage, but since they're such a more vigorous root system, whether we could count on them to give us more of a weed-free period. Critical period for weed control in grafted and non-grafted watermelons just shows you the example of that weed-free period. She did 3 rootstocks, and well, she did 2 rootstocks basically, an interspecific hybrid, which is a carnivore, and then the Casaco, which is a wild watermelon type. This just gives you the critical period of weed in grafted versus non-grafted on marketable yields. And just to show you what that weed-free period is. See how crucial that is. If you go back a slide, you'll see how crucial that period is, where the first few weeks of growth are real critical to the actual production of watermelons. This just gives you the effects of Palmer amaranth, which is becoming a real problem in our watermelon production in the use of grafted plants and how that affects. Summary, with the goal of 95% weed-free yields, the critical periods are as follows. With exclamation alone, it's between 15 and 18 days after transplant. Exclamation grafted onto carnivore, it's between 13 and 18 days after transplant. An exclamation on Casaco, it's from 2 to 18 days after transplant. In other words, we're not really getting much of an advantage of grafted plants. Watermelons are very high valued crops. And so you really need to keep the plants clean as possible without any weed pressure until about 32 days after planting. That's the weed-free period that weeds need to be kept under control. Now, the last I want to talk to you about is the organic production of watermelons. It is becoming more and more of a possibility of organic watermelon production, especially in the southeast of the United States and on the coast. And Dr. Brian Ward at Clemson University has been heading up that study. Watermelons that are grown organically are sometimes worth as much as 5 times to even higher amounts of actual returns on watermelons if grown organically. Brian has been studying the bee population, the native bees and honeybees related to watermelons, especially with grafted watermelons. And he's found one type of a bee that's less attracted to watermelons than other bees, but in generally, it didn't really matter whether it was grafted or not. The bees still came, whether they were wild or native bees. He also has been very high on Carolina Strongback because they've outperformed all other rootstocks in the grafting. The vigorous grafting rootstocks seem to really help his organic production. Grafted watermelons typically outperform non-grafted plants. Crown set of watermelons was increased using grafted plants. And he also uses wildflowers to enhance the movement or bring in more of the wild bee population or the native bee population in our area. Most important in his work, he has come up with a way of growing grafted watermelons organically using a clover between the plastic strips. The regal ladino grazing clover inhibits all weed pressure and limits all need for any hand type of weeding, which is a real problem in organic production. And that's where we really use a lot of our weed problems. Remember that 32 days, 35 days after transplant is so critical? Well, with the regal ladino clover, he can seed a field and then cut out his plots. And the regal ladino clover is so vigorous that it pretty much reduces the weeds. And this is what I wanted to show you, is this shows you the field of organic production that he grows with the wildflowers on the side. But that clover in between the plastic strips holds down all weed pressure. And therefore his yields are very comparable to any of our commercial fields. The diseases have been a problem and continue to be a problem, but I think he's getting a handle on getting controls of those. But he's getting a lot of interest from organic growers as far as production goes. Now I want to talk to you a little bit about pepper rootstock that we've been working on. It'll probably be covered in the genetic working group, but in case it's not, I just wanted to, because we're pretty excited about this in the solanaceous working group, about a Phytophthora, a crown and root rot resistance to Phytophthora in peppers. The objective was to develop new Phytophthora crown and root rot resistant rootstocks for grafted pepper and evaluate rootstocks grafted in the greenhouse and heavily infested field and evaluated on a commercial pepper field. This just shows you that the lines that we developed from the PI line 201232 that we were able to acquire, and then we've been able to select out of that. E1 rootstock has been released earlier from this PI line, but the discovery of E3 rootstock is one that we're very proud of because it's a lot more vigorous and contains a high level of resistance to Phytophthora. We're calling the E3 hassle-free and soon to be released. We have an abundance of seed of that if any of you would like to try it. The grafting of pepper plants is very, very simple to do. But this just shows you, these next series, it just shows you the resistance. Dr. Kosek has a number of fields that he tests this in that are very heavily infested with Phytophthora. And these seem to be the only thing that holds up. This just gives you the Jupiter grafted on Jupiter and Jupiter non-grafted versus the Jupiter on E3. This just shows you the pressure that he has with Jupiter alone on plastic and Jupiter grafted onto E3. This year we did a huge grafting grower trial on 2020 on Weir's Farms Incorporated in Willard, Ohio. We used the cultivar Scion, the Scion material 3255 from Cinemas. It's a very beautiful, high-yielding pepper with bacterial resistance, but it doesn't have particularly good tolerance or resistance to Phytophthora. The two rootstocks, E1 and E3, just shows you the picture of the two. E3 produces almost twice as much seed per fruit as does the E1. And that's just an advantage to those that may be producing that seed. We did this at Weir's Farms, planted in April. Seeded in April at the coastal station in Charles, grafted it in May. And the end of May, we transplanted out at Hack Farms, a branch of Weir's Farms Incorporated in Tifton, Ohio. This just shows you the plot that we did. We delivered the plants and they grafted them all. Then we went through in July. I flew up and we evaluated those plants. We did 10 plants per rep. We did 5 replications, randomized complete block, randomized design in there, and harvested those. I just want to show you preliminary information. Notice that the non-grafted, the number of fruit per plant versus the ones that were grafted on the E1 and E3. So you also look at the size difference. The ones that the market is after are the 3 inches, 3x3s. Those are, and if you look at the difference in those as well, you'll see a substantial difference. The grafted plants were much shorter, and the compactness of the plant produced more 3 and 3.5 inch. So we're really excited about the results of this. Whether it becomes economically feasible to use grafted plants in a pepper production is still out there. But if it becomes more of a serious problem, this is definitely an option. So E1 and E3, E3 is going to be named hassle-free, high resistance to crown rot, as well as greenhouse field resistance. Those resistance proved to be very beneficial, and the quality of the fruit was excellent. So we're looking forward to further tests on that. This concludes my talk as far as the Cucurbit working group is concerned. If you have any questions, feel free to give me a call. You can email me at rhassel@clemson.edu, and I can direct your call to wherever those scientists that may be interested in finding out more information about our working group. Thank you again for your attention, and look forward to hearing from each of you in the near future.